the Ministry of Higher Education in Jordan, in addition to 9 other local and regional awards. So please welcome with me Dr. Mu'tasim Tahi.
Thank you very much. I will be talking today about the development of novel QSAR techniques to enhance success rates of ligand-based and structure-based computer-aided drug design. The research presented here is being conducted at the University of Jordan. Drug discovery and drug design can be divided into two methods, either structure-based, whereby you know how the binding pocket looks like or the lock looks like. And you have also this is how the binding pocket, for example, this is the receptor, and in this case you design a ligand that fits the lock of the binding pocket. This is structure-based. And if you don't have any idea about the binding pocket, you can go for ligand-based, whereby you test different keys or different ligands, and you try to find the common features among them in order to find a good key to open the lock. One method in ligand-based design is so-called pharmacophore modeling. Pharmacophore is what would be, if you take a group of ligands that are known to bind to certain receptor, you put them against each other to find the common features among them. You can build a pharmacophore model or a binding model. For example, you might have an aromatic region here, hydrogen bond forming group here, hydrogen bond donor forming group here, with certain distances. Such a pharmacophore is quite efficient as a search query to find hits. Such hits can be active in vitro. What we did additionally, we combined the pharmacophore exploration process with the QSAR modeling process in a novel way of modeling. By doing so, we could find several pharmacophoric models that describe the binding of a particular compound within the binding pocket. So by doing so, we could actually identify several binding poses for the same molecule. And in this way, we can monitor how the molecule binds and moves within the binding pocket. As you can see in this picture here, the compound moves in a certain way inside the binding pocket, and this was discovered by just pharmacophore modeling and QSAR analysis. We applied this approach for several examples. For example, in this case, we applied it against the target glycogen synthase kinase, which is an anti-diabetic target, and we could isolate several drugs that bind this target with nanomolar inhibitory affinities. You can see also the pharmacophoric models that we found correspond with the crystallographic structures of the binding pocket. Same applies for this target, hormone-sensitive lipase, which is also involved in diabetes. And we could also find two binding models and a good QSAR model to explain the bioactivity of the hits. And hits were tested, and we could identify micromolar and submicromolar inhibitors from in vitro testing. Some of the inhibitors are known drugs, like this one, naproxen, and some of them are agrochemicals, like bifenox, which is a herbicide. So we could identify several inhibitors of this target using this approach. Similarly, the target dipeptidase 4 was also analyzed by pharmacophore modeling and QSAR analysis, and we could identify two binding models that we implemented as a search queries to find hits. One of the hits was significantly potent with IC50 equals 1.2 micromolar after in vitro assay. Similar approach we conducted for the influenza neuraminidase enzyme in an attempt to find anti-influenza drugs or treatments. We modeled the ligands for this target, and we could identify 5 binding modes. We used the 5 binding modes to capture hits that we tested in vitro, and 4 of the hits were rather potent according to in vitro assay. Similarly, similar analysis has been conducted against estrogen receptor beta and an anti-cancer target, heat shock protein 90. Heat shock protein 90, we captured several drugs that have nanomolar inhibitor activity, and they actually had anti-cancer properties as well. Some of those drugs, as you can see, are anti-malarials, like amidoquine and primaquine, and some are anti-asthmatic drugs. And they were having anti-cancer properties, unexplained anti-cancer properties. Now we know their properties, anti-cancer properties are related to inhibition of this target. And we could discover this behavior by pharmacophore modeling followed by QSAR analysis, followed by in silico screening and in vitro assay, as you can see. This is another anti-cancer target, CDK1, which we modeled as well using the same approach, pharmacophore modeling followed by QSAR analysis, capturing in silico hits and testing them in vitro to find bioactive compounds. We also started to synthesize compounds guided by our pharmacophore models. In this case, we built some anti-dyslipidemic agents, inhibitors of this target, cholesterol ester transfer protein, which is involved in dyslipidemia. And we could find 2 active compounds against this target in the micromolar range. Here, we also targeted the enzyme beta secretase, which is involved in Alzheimer's disease. And we built several, as I said, many pharmacophore models. We allowed QSAR analysis to capture the most potent or the most successful pharmacophores. And then we use the pharmacophores to guide synthesis of beta secretase inhibitors. One of the inhibitors that we synthesized produced 1 micromolar anti-beta secretase activity, and it was rather potent, as you can see. Here, similar analysis we conducted against beta-galactosidase, which is an anti-diabetes target, as well as beta-glucosidase, another anti-diabetic target. And here, against antifungal target, which is the myristoyl, N-myristoyl transferase. And we could also identify antifungal agents in this way. Okay, similarly, we also targeted the enzyme glycogen phosphorylase, which is also involved in diabetes, and we could capture active hits against target. And also we synthesized new compounds that fit the same pharmacophore, and therefore they were found to be active as well. So we synthesized several inhibitors of this enzyme as well. This is not clear, but 2 of the synthesized compounds were actually sub-micromolar or at low micromolar inhibitory activity. Similarly, we targeted the enzyme calcium calmodulin dependent protein kinase 2, which is an antihypertensive target. We built hundreds or even thousands of pharmacophoric models, and then we allowed QSAR analysis to pick the best models. We used them to search in silico databases for active hits. We captured many hits. And we also used the model to synthesize active compounds. One of the most active compounds had an IC50 of 0.15 micromolar, 150 nanomolar. Okay, similar analysis also was conducted against rho kinase, which is another antihypertensive target. We captured as well active inhibitors of this target. And finally, we carried this analysis as well against nitrous oxide synthase. Nitrous oxide synthase is a target involved in cancer as well, and inhibiting this target should stop angiogenesis. And we actually found extremely potent inhibitor of this target with an IC50 value of 15 nanomolar. Maybe it's not clear here. Quite potent compound, even not 15, 1.4 nanomolar inhibitor. 1.4 nanomolar. And this is the target, how it fits. It forms actually a covalent bond within the INOS target. Okay. A similar also approach was conducted against beta secretase transition state analogs. And we captured hits against target. Now, the other thing that we are doing in my research group is to carry out validation of structure-based design using QSAR technique, a new novel QSAR technique. If you use crystallographic analysis or docking, you will probably suffer from some issues in the discovery process. First, the process of crystallization causes loss of 3D structure of the protein. The crystallographic resolution is not always optimal. Water molecules can either be removed or kept. You never know what will happen upon binding. The same also regarding protons of ionization. Do we have ionized groups or unionized groups? It's not clear. Protein is flexible and you might have induced fit. And docking doesn't calculate entropy. So overall, the process of docking as a method for structure-based design is not an accurate approach and it requires validation. So what we did, we looked at the validation techniques that are found in the literature, and we find each of those techniques has a problem, an inherent problem in it. For example, self-docking has a problem of induced fit. Rock analysis has a problem of assuming inactive decoys. 3D QSAR methods like COMFA has the problem of being very time-consuming. So we introduced our own method here. We did use COMFA analysis for validation. However, as I said, we introduced our new method here, which we call docking-based comparative intramolecular contacts analysis. This is a novel approach, and this was an award-winning approach. Recently won the award of the Ministry of Higher Education in Jordan. This is a novel approach based on the number and quality of contacts between the docked ligands and amino acid residues within the binding pocket. It evaluates docking by assessing the correlation between ligands affinities and their contacts with binding site. And the optimal models, DBCK models, can be translated into valid pharmacophores. So what we do here, we start by having a binding site of a protein. We dock all our compounds inside that binding site. And then we calculate the distances between different amino acids within the binding site and the corresponding docked compounds. We then generate something called distance matrix, whereby those molecules that are in contact with certain amino acids are given one binary value of 1, while those which are not in contact are given binary value of 0. Then we carry out genetic algorithm-based summation of QSAR analysis to identify the contacts that really differentiate between actives and inactives. Then we can convert those contacts into a pharmacophore model. The pharmacophore model can be used as a search query. We use this approach to capture inhibitors of immunohistoid transferase, antifungal target, and glycogen phosphorylase, antidiabetic target, as examples of this novel approach. So we have, as you can see, several hits. Some of them are rather potent. Similarly, we applied the same approach against heat shock protein 90, which is an anti-cancer target. And we also could collect a very nice pharmacophore model that we implemented a search query to capture quite potent inhibitors. Okay. So these are the hits that we capture using this novel approach. Currently, we are interested in building pharmacophore models using molecular dynamics simulation. If you simulate, the theory is quite simple. Ligand receptor recognition and binding are mediated by a limited number of critical interactions, while crystallography complexes usually exhibit more than 25 interactions, many interactions. So we can use molecular dynamics to differentiate critical from redundant interactions, and then convert critical interactions into pharmacophores. This is the workflow that we are doing now, and we hope it will be successful, because the basic idea is quite reasonable. I think it will be successful. Thank you very much. Any questions?
Thank you very much, Dr. Hassan, for this very interesting lecture. I've got actually two questions. The first one about the binding modes. The binding modes. So you usually have multiple binding modes in order to generate the pharmacophore and screen them. So are they very different from each other, or just there are little differences?
Sometimes it can be quite significantly different. Yeah.
All right. So
what we noticed on our previous work, that if a particular molecule is potent, it does have the ability to assume many different pharmacophoric modes inside the binding pocket. The more potent the compound is, the more pharmacophoric binding modes it can assume in the binding pocket. So, yeah, it looks, sometimes you might have really significantly different binding modes.
The second question about the inhibitors themselves. So, you know, there are some of them could be like promiscuous inhibitors, which actually inhibit the protein by non-specific mechanism.
Yeah.
So like
we normally test for promiscuosity.
Yeah, so what kind of test?
First of all, promiscuous behavior appears from atypical dose-response relationship. If you have at low dose, you have certain activity, and then the activity jumps and goes down again, and then jumps and goes down. It doesn't fit a proper dose-response curve, then this is probably a promiscuous compound. And we usually filter those carefully. I usually make great efforts to check for the validity of my hits.
So do you try detergents?
We also sometimes try doing the assay in albumin. Sometimes we also test the time dependence of inhibition. If you leave it for a long time or a short time, let's see what is happening. The minute that we suspect promiscuous behavior, we check for it.
Thank you very much.
Thank you for an excellent talk. I have a question about your INAS inhibitor. Your INAS inhibitor was a covalent binding inhibitor. And I was wondering if modeling covalently binding inhibitors presents any kind of challenge for you when you're looking, because I know that these covalent binders now are really important, so is that a challenge?
In fact, we usually avoid covalent binders in our modeling training sets, because covalent binders usually are more potent than should be based on pharmacophoric recognition. So what happened with this compound, it was actually captured as a reversible binder, but then upon testing, we found it to be extremely potent. So we did some more testing, like dilution and checking the activity in situ. And we found the compound is actually irreversible. It inhibits the enzyme, and the inhibition does not stop upon dilution. So and then we hypothesized that it formed a covalent bond due to the 5-membered ring, the hydrogen bonded 5-member ring that you saw. It's like a carbamate type group. Carbamates, as you know, is well known for forming covalent bonds with the respective targets.
Okay, we'll take the last question.
Dr. Matos, thank you very much for the interesting lecture. Just I was wondering, in your new approach about 2001, do you check the X-ray crystallographic structure before you start 2001? Because I know
Check for what?
The X-ray crystallography. The
Crystallography.
structure itself.
Yeah.
Because I noticed from my experience that some published structures, they have some difficult, something wrong in it.
Yes.
Do you check that?
We normally check for crystallographic structures before doing any docking or any studies that we do. If we find too many flexible regions, we usually ignore such structures. If the resolution is bad, we ignore it. We usually filter it before doing some work with it. Usually. More questions? So,
Thank you. But the last question, 1 minute. Can we estimate the size, and does the size matter? The size of the molecule or the compound?
can we check size? Yeah. You mean molecular weight of the compound?
No, the size, I mean, is nanometer or the size?
Oh,
How
the
big
size
is
of the compound. Well, compounds are usually in the angstroms dimensions.
And
So we don't check for the compounds. The pharmacophoric arrangement that we look at is separated by certain distances. Each feature is separated from other features by certain distances. And usually distances are in angstroms.
But I'm talking about the chromophore, the whole structure. Does it, I think we can get it in nanometer or?
No, smaller.
And it's wrong.
Mm.
Okay, thank you.
Okay, thank you, Dr. Matos. Actually, it's a pleasure to welcome and introduce our second speaker this session, Dr. Amanda Bryant Frederick. Amanda got her PhD from the University of Heidelberg, Germany, in pharmaceutical chemistry, and she got her postdoc training at the University of Basel, Switzerland, in physical and organic chemistry. Amanda actually is one of the recipients of the National Science Foundation Career Award. She's associate professor in medicinal chemistry in the College of Pharmacy, University of Toledo, Ohio. And her talk today is about the chemical toxicology of endogenously generated reactive oxygen species and their role in cancer etiology. Welcome again, Dr. Amanda, and you have the
Thank you very much. It's very nice to be introduced by a member of the family, so to say. So I really appreciate that introduction. So today I'm going to give you a little short introduction to what we actually do in my laboratory. My title actually talks about chemical toxicology of endogenously generated species, reactive oxygen species. But I'm going to talk a little bit about also radiation-related reactive oxygen species as well. So this is my always introduction slide because I have to get people to remember some of these structures. What we're talking about when we're talking about reactive oxygen species are all of these small, highly reactive species that are generated. On this side, what you see are ones that happen to come from the fact that we breathe oxygen, of course, and our body has to metabolize it. So you see hydrogen peroxide here, you see singlet oxygen, you see the hydroxyl radical, which is, of course, the major oxidant, and then you see down here the oxygen radical. What you see over here is hypochlorite. Hypochlorite is, of course, coming